A warm welcoming everyone. I'm Poovili from 3rd BSc Biochemistry. I'm going to say about plasma lipids and lipoproteins. Plasma lipids. Since lipids are insoluble in water, they need the help of carriers in plasma. Therefore, they are complexed with protein to form lipoprotein. The protein part is called apolipoprotein. It is abbreviated as LP. Types of plasma lipids. Based on the chemical structure, plasma lipids are four types, namely cholesterol, fatty acids, triglycerides, and phospholipids. Now we are going to see about cholesterol. Cholesterol. It is a steroid precursor to bile acids. It is a steroid hormone. Cholesterol level in body is equal to 150 to 200 mg per dl. It is synthesis in liver and intestine. Free cholesterol in bile. Bile acids are important in absorption of cholesterol. Fatty acids. It is a straight chain carbon compounds in plasma. Free fatty acids are carried bound to albumin. It is an immediate source of energy. Yield large quantities of ATP. Exists production of ketone bodies. It is known as ketosis. Next, triglycerides and phospholipids. Triglycerides. Fatty acid esters of glycerol containing three different fatty acids. They are transported as lipoproteins. After hydrolysis, reesterified and stored as triglycerides. Phospholipids. Complex lipids that reassemble triglycerides but contain phosphate 1 plus nitrogen base in place of one fatty acid. Lipoproteins. Lipid plus proteins is called lipoprotein. It is synthesis in liver and intestine, modified after secretion by enzyme catalyst reaction. Lipids are classified based on their activity that reflects the size. Next, we are going to see about classification of lipoproteins and their origin and their function. Classification of lipoproteins. Lipoproteins. Kilomicrons, VLDL, IDL, LDL, HDL. Kilomicrons. Transport exogenous lipids from intestines to all cells, large, triglyceride rich. VLDL. Very low density lipoprotein. Transport endogenous lipids from liver to cells, large, triglyceride rich. Ideal. Intermediate density lipoprotein. Transient intermediate formed during conversion of VLDL to LDL, undetectable in normal plasma. Cholesterol plus triglycerides. LDL, low density lipoprotein formed from VLDL, transport cholesterol to cells, contains mostly cholesterol. HDL, high density lipoprotein, transport of cholesterol from cells to liver. Their origin and functions. Kilomicrons, their origin is intestine, function is absorption of dietary fat. Kiloremnants, their origin is plasma, its function is to deliver dietary fat to liver. VLDL, its origin in liver, its function is deliver triglycerides from liver to other tissues. Ideal, its origin is plasma, its function is to initial product of VLDL catabolism. LDL, its origin is plasma, its function is cholesterol transport. HDL, its origin is liver intestine, its function is removal of excess cholesterol from tissues and lipoproteins. It's a function and origin of lipoproteins. Thank you everyone.